Hello, everybody, and welcome to another distance learning episode here. Hope everyone's doing um, as well as possible, and enjoying at least a little bit of something new, something learning stuff. And the podcast facts came in a bunch more, and so we'll definitely be adding those today to the new podcast. They seem to be pretty popular, and so I'm glad that's good. Our goal for today for Myosis is to do a quick little review of what we talked about yesterday and then move forward with the process a little bit. So I believe if all went well, I should have my screen up here. And we are talking about Meiosis. My spelling did not get any better over the last couple of weeks. Sorry about that. And this process, which it is a process of producing 4 genetically different reproductive cells. Now, notice how that's different than mitosis, where you are getting 2 genetically identical cells. Those are the body cells. In meiosis, we're producing 4 genetically different reproductive cells. Right. Another new term we learned yesterday was homologous pairs. Think about yesterday. What was that? That happened in Prophase 1 when the genetic material was able to leave the nucleus because the nuclear membrane broke down. They didn't just float around on their own. They floated around with their homologous pair, and that was pairs of chromosomes that are genetically similar. So they're genetically similar in size and shape. Chromosomes are not all the same exact size. So you'll have ones that are slightly larger, slightly smaller than others, different thicknesses. If you go to Google and just type in human chromosomes, and look at the images, you'll see what I mean. They are different sizes. So a homologous pair is a pair of chromosomes that are genetically similar. And yesterday a gamete, that was at the end of cytokinesis 1, the first time it happened, you end up with 2 gametes, and those are 2 separate cells. In mitosis, we call them 2 daughter cells. When they split and went apart, they were genetically identical daughter cells. Those gametes are not. Gametes are 2 genetically different reproductive cells. So in the case of the salmon yesterday, or birds or dogs or cats, or you and your brother and sister, you are not genetically identical unless you're twins. You are not genetically identical. You're going to look similar-ish maybe, but because of that crossing over, and that is when those chromosomes actually when they're lining up with their homologous pair, sometimes they cross over and switch genetic material. And that is a physical crossing of chromosomes and exchange of genetic material. And feel free to pause this video at any point and make those notes there. And that's pretty much where we got up to yesterday. And if you want to pause it and take those notes in your notebook, go ahead. And I'll assume you are back, or you're just going to keep going. So let's look at here. So if I was starting meiosis, think to yourself, what should we label this here? What should we label this first step? And hopefully you're thinking interphase, just like for mitosis. And what's going to happen here? What are we going to get? We're going to have duplicating the DNA, duplicating organelles, right? Duplicating cytoplasm. Right? So we can put some organelles in here. Whoa, I don't know what that is. Let's say it's a mitochondria. Put another one. All right, we'll put some of these guys in. So we're just showing that we're duplicating all of the organelles in here. We don't want to do that. And we'll put a nucleus. And we'll put our genetic material in. Some of them like that. Right? And then some like the X. Okay, interphase. And we could even say it is growing and developing because it's getting bigger, but it's also becoming more complex. All right, so if you want to pause the video and make your meiosis interphase, go ahead and do that. Next up, meiosis. It's going to be prophase 1, because we're going to go through prophase twice. Okay, so make it nice and big. We are not going to draw all the organelles again. All the organelles are duplicated. All the DNA is duplicated. All DNA duplicated. Which means then we have how many chromatid and chromosomes. We started with 46 chromatid, and now we have 92 chromatid. All organelles duplicated. What happens to the nuclear membrane? Right, it begins to break down. Now when the nuclear membrane starts to break down, how are the chromosomes moving out, and what are they starting to find? They're starting to find their homologous pairs. We're going to put our nuclear membrane breaking down, and we're going to make them, this one, about the same size and shape as this one. So we'll try to draw it the same way. Good. And we'll draw one over here bigger. And then its homologous pair will also have to be bigger. We'll start to line up with our homologous pairs. And we'll put one here. And then we're also going to put some crossing over. Whoops. That wasn't so good. Some crossing over. Right here is where that genetic material is being transferred from this chromosome to that one. And we'll put one still inside. Of course, we're not drawing all of them This should be a homologous pair about the same size and shape. And I did a pretty good job of that. Right. Now there's two things that start to form. Remember what they were? I hope you do. They were the spindle fibers and the centrioles. And they, just like before, they start to form at the poles. So we'll draw our centrioles and spindle fibers starting to grow. All right, that's prophase 1 in meiosis. So a couple of differences. Homologous pairs are forming, and there's some crossing over happening. Okay? Everything else is the same. All right. So what's next? Meiosis, prophase, and hopefully you're saying out loud to your cat, to your brother or sister, you're saying anaphase. No, you are saying metaphase 1. Okay. And then we'll put a cell. And that M tells us that our homologous pairs are lining up in the middle. This time, instead of, well, that's not a very good homologous pair. Try and make it better. There we go. We'll do a little one. Another pair, a little partner. And we'll do a crazy one. And do, oh boy, I gotta try to pair that up. Here we go. There we go. Okay, we'll put our centrioles and our spindle fibers attaching. Now, last time in mitosis, if you remember when we split this, we split these X's apart. But we're not doing that in meiosis metaphase. We are splitting metaphase 1. We are splitting the homologous pairs away from each other. But that will be next. Remember, they are attaching to the centrioles, which are in the center. All right, homologous pairs lined up in middle. Spindle fibers attaching to centromeres. All the organelles are still here, and we're ready to move on. Where are we going next? Hopefully you're saying out loud or in your head, You're saying you wish I could type better? Me too. Metaphase, anaphase 1. All right, let's do our words first. What's going to be happening here? Homologous pairs are being separated. Okay, and how many are going each way? Okay, so we have 96 chromatid altogether, and they're separating evenly. 92 chromatids, so we should have 46 chromatid going one way and 46 going another. So you have 46 chromatid going towards one pole and 46 chromatid going to another. All right. And let's draw. Okay, and this is his pair here. going that way. Little guy. Little guy. And we'll do our crazy backwards K looking one. Crazy backwards K looking one. Spindle fibers attached. Okay, so we should have 46 chromatid. 46 here. And 46 there. 46 going this way. And 46 going this way. For the total of 92. Hey, anytime you want to pause the video, you can pause it and make your drawings. Okay, next up, after anaphase. Ooh! You know what we forgot to do? I bet you probably already knew. We forgot that cytokinesis begins. We forgot about that. Well, I forgot about that. Can I erase some of this? That's a good question. I bet you I can. Oh yeah, we'll put some whiteout on it. Because we have to get cytokinesis beginning. Sorry if you're erasing at home. I really am. Get that cleavage furrow going. That's not bad. Well, it's not good either, but it's not bad. All right, cytokinesis begins in anaphase. I tell you, some things don't change. All right, meiosis. We're going to have to hand draw this one. This one is telophase 1. And we need to draw a deeper cleavage furrow. Complete with sound effects. Okay. We are going to have returning. What's returning? Our nuclei. Because what we don't want happening is our chromosomes getting away. They are trapped in here. And the nucleolus is reforming. And we'll put a over here. Full X's. Full X's over here. Okay, on this side, we have 46 chromatid. Over here. And we have 46 over here. On that side. Cytokinesis continues. Nuclear membrane forms. Nucleolus forms. Centrioles and spindle fibers go away. Spindle fibers, what? They disappear. Disappear. Okay. Looking pretty good. All right. On to. Oh, yeah, cytokinesis continues. I want to forget about that again. All right. Meiosis. Cytokinesis. We're going to draw two genetically different gametes. And that's what we're calling these. They are not identical. Because of that crossing over, some of the homologous pairs went one way and some went the other. They're going to be different. Put a few chromosomes in there. Oops. That one's leaking out. Okay, and there should be 46 in this one. And 46 in this one. Now, if you're thinking ahead, I said in meiosis we go through this twice. So we're going to go through this whole process one more time. And when you think about that, this 46, this one is going to split into two that have 23 each. This one will split into two that have 23 each. And at the end, we're going to end up with 4 cells, each of them with 23 chromatid in them. Okay, that's where we're headed. Okay, I think that's enough for today. That's a lot of little review there and you making your notes. So I think that's good. Tomorrow, we will go into meiosis 2, like the second time through it. And then we'll look to see how we end up with 4 cells, 23 in each. If you want to go ahead and look at the videos up to that point on our website, there's a meiosis one. And you can watch up to the point where we are at, or you can watch the whole thing and get a preview of what we're doing tomorrow. I am going to work on the Science Facts podcast and get that out in a few minutes as well. All right, I hope everyone is doing well, staying healthy, staying safe, and I will see you in the podcast in a few minutes. And all right, talk to you later. Bye.